Okay, so the topic of this video is the cellular division process known as meiosis. I challenge you to come back to this animation right here at the end when you're done and you think you have a good understanding. Come back to this animation here and try to talk out the events that are happening in this animation. The animation is only 15 seconds long or so, but try to talk out what's happening once you think you have a good understanding and just see if you can talk your way through it. So let's go ahead though and get started. Okay, so let's go ahead and get started. First of all, the definition of meiosis. Meiosis is the process where the chromosome number of a cell will be reduced by half. The picture here, I see 2 chromosome 1s and 2 chromosome 8s and 2 chromosome 12s and 2 chromosome 18s. This is a karyotype of a diploid cell. So cells that undergo meiosis are going to start diploid. So a human cell that undergoes meiosis will start with 46 chromosomes. However, when meiosis is finished, read the definition, it's the process where the chromosome number is reduced by half. Watch how the picture changes. When meiosis is finished, the cell will end haploid. There's only going to be 1 chromosome number 1 and 1 chromosome 9 and 1 chromosome 12. In humans, a cell that undergoes meiosis will start with 46 chromosomes but will end with 23. Meiosis makes haploid cells. So meiosis is what makes the haploid cells called gametes. So meiosis makes haploid egg cells and meiosis makes haploid sperm cells. These again are called the gametes. And when meiosis is finished, the end result will be not just 2 cells like mitosis created, but 4. I already mentioned the cells are going to be haploid, but meiosis is going to create 4 cells. Here we see a picture of 4 cells that were created by the process of meiosis. Okay, so let's get into the first step of the cell cycle. If you recall, interphase, it's not a part of mitosis, and interphase is not a part of meiosis either. But interphase is a part of the entire life of the cell, known as the cell cycle. Before we get started, I want to put a note in the upper left-hand corner. If you look in this animation, or in this cell, there are 6 strands of DNA in the cell at the start of this animation. 3 strands of DNA in black, 3 strands of DNA in red. So I want to put that note in the upper left-hand corner just as a reminder. At the beginning of this animation, there were 6 strands of DNA in this cell. Well, let's go through the stages of interphase. Now, in my previous video, I also explained the steps of interphase, and it's the same process as my previous video. Now, I mentioned in the previous video that interphase is subdivided into 3 categories. Subcategory number 1 is called the G1 stage. The cell grows, and it makes sense. If the cell is about to divide and split, it makes sense that it should grow and get bigger. Also mentioned that additional organelles are created, additional lysosomes, additional vacuoles, additional ribosomes. And again, it should make sense. If the cell is about to divide and split, it should make sense to make more parts. That way, every cell that's created will have its own supply of parts. Then there's the second stage of interphase called the S stage. The S stands for the word synthesis. And the word synthesis just literally means to make. So in the synthesis stage, the S stage, the cell is going to make something. And what it's going to make is the loose, stringy version of DNA called chromatin. In my animation, those 6 pieces of chromatin are going to be duplicated into 12. In a human cell, humans have 46 pieces of DNA, 46 strands of chromatin that would double and duplicate into 92. But in this animation, we're going to follow a cell that had 6 strands of DNA, but it just doubled into 12. Then there's what's called the G2 stage. It's the second growth stage, and the cell will grow again, get larger. After all, it's about to split and divide. So the interphase stage that we described in the previous video is really the same as in this video. Well, now let's go ahead and jump into meiosis. Here we go. Okay, so let's actually get into meiosis now. The first stage of meiosis is called prophase number 1. there eventually will be a prophase number 2, but let's not get too far ahead of ourselves. Prophase number 1. First of all, in the animation, the chromosomes are going to form. The loose linear versions of DNA called chromatin are going to wind up and condense into these X-shaped chromosomes. I'll explain in a few moments why I labeled some chromosomes, mom number 1 and dad number 1 and mom number 2 and dad number 2. I'll explain why I did that in a few minutes. Another thing that happens in prophase number 1 is the nucleus is going to dissolve. So these two things that I just mentioned, the chromosomes forming and the nucleus dissolving, this happened in prophase of mitosis as well. So prophase of mitosis and prophase number 1 of meiosis are shaping up to be pretty similar so far. However, a couple of important events also occur in prophase number 1 of meiosis. Let's talk about those now. So the two events that take place during prophase number 1 of meiosis that are unique to meiosis is the event called synapsis, which is the pairing of homologous chromosomes. And this is why I labeled the chromosomes from mom or from dad. What's going to happen is dad's chromosome number 1 will pair with mom's chromosome number 1. The two chromosome number 1s will pair up side by side. The two chromosome 2s will pair up side by side. The two chromosome 3s will pair up side by side. In this animation here, I made all of dad chromosomes that you inherited from your father, I made them all red, and I made all the chromosomes you inherited from your mother black. So in the animation, you saw synapsis occur. And now what we have are these groupings of two chromosomes. These are called tetrads. These groupings of chromosomes are called tetrads. And the reason for the name tetrad, if you know the prefix tetra is a prefix that means 4. Well, if you recall that one chromosome is made from two chromatids, therefore these two chromosomes of a tetrad are actually 4 chromatids. And that's why they get the name of a tetrad. And so this happens during prophase number 1, and this event is called synapsis. Well, there has to be a reason for this to happen. Let's see what that is. Once the two chromosomes have paired up next to each other, once the chromosome number 3 that you inherited from dad pairs up with the chromosome 3 you inherited from mom, an event called crossing over occurs. And I think I can visually show crossing over better than I can explain it. So let's go ahead and let me show you what I mean by crossing over. Let's zoom on into this tetrad that I have highlighted here. And here we have, again, in red, the chromosome number 1 that you inherited from your father, and in black, the chromosome number 1 you inherited from your mother. The definition of crossing over is simply the exchange of genes. Watch what happens. During the process called crossing over, chunks of chromosomes will break off and reconnect. So a chunk of dad's red chromosome broke away and reconnected to mom's. A chunk of the black chromosome that you inherited from your mother broke off and connected to the chromosome you inherited from your father. Well, let's zoom back out and take a look at the entire cell. So now that we've zoomed back out, you can see the tetrad on the left has experienced crossing over, but the two other tetrads have not. So there you just saw crossing over occur, and here it is again. So now that crossing over occurs in each of the tetrads, what this does, I can't stress this enough, the importance of crossing over is it creates genetic diversity. Eventually, meiosis is going to create sperm cells and egg cells, and because of crossing over, each of them is genetically unique. At the end, they're all the cells that are made by meiosis are going to be different, genetically different, and you'll see that as we continue on. So as we move on into metaphase number 1 of meiosis, the homologous chromosomes, again the tetrads, are going to align along the cell's equator. If you recall, the spindle fibers will grow from centrioles, attach themselves to the centromere, and pull the chromosomes to the middle or the equator of the cell. Okay, as we move on into anaphase number 1, The chromosomes are still in the middle of the cell. That would happen during metaphase number 1. But in anaphase number 1, the tetrads are going to be separated. Now I've got 3 tetrads in this animation. I have a tetrad on top, a tetrad in the middle, a tetrad on the bottom. So in a moment in my animation, remember that a tetrad is made from 2 homologous chromosomes. They're going to be pulled to opposite ends of the cell. So the tetrad on top, in a moment, one homologous chromosome will be pulled to the left by spindle fibers. One homologous chromosome will be pulled to the right by spindle fibers. The tetrad in the middle, one homologous chromosome of the tetrad in the middle will be pulled to the left. One homologous chromosome of the tetrad in the middle will be pulled to the right. And finally at the bottom tetrad. Spindle fibers are going to pull one homologous chromosome to the left, one homologous chromosome to the right. Here we see in the animation the tetrads are being separated. So in anaphase number 1, the tetrads are separated, and that leaves one homologous chromosome at each end of the cell. So if we look at telophase number 1 of meiosis, well, cytokinesis is going to divide and split the cell, and there you can see cytokinesis dividing and splitting the cytoplasm. We now have the formation of 2 cells here. Sometimes the nucleus will regrow, sometimes it doesn't. In this animation, I'm going to have the nucleus regrow around the chromosomes that you see in those 2 cells. And each cell is going to contain one of the homologous chromosomes, but note that the chromosomes are still full and intact. They haven't been ripped apart yet into chromatids. That will happen, just not yet. And so in humans, if these 2 cells were humans, there would be a cell on the left with 23 full chromosomes and a cell on the right with 23 full chromosomes. But this isn't the end. This is only the end of meiosis number 1. Now we go into meiosis number 2. And meiosis number 2 begins with prophase number 2. By the way, that's a Roman numeral 2. That is not prophase 11. It's prophase number 2, Roman numerals. And so Prophase number 2 and Metaphase II and Anaphase II and Telophase II, they pretty much follow the same steps as mitosis. So the nucleus is going to dissolve, the spindle fibers are going to form. There you see the nucleus dissolving. One thing I want to point out is that notice how we went from telophase number 1 into prophase number 2. There was no second interphase, and that's really important. If you think of the point of meiosis, the point of meiosis is to reduce the amount of DNA by half. So that's why there is no second interphase. If there was a second interphase, there would be a second S stage. And what happens during the S stage? During the S stage, the cell makes more DNA. Well, the purpose of meiosis is to get rid of and lose half of the DNA, not to make more. So that's why there's no second interphase. And so as the cells move on through metaphase number 2 of meiosis number 2, the remaining chromosomes are going to align at the cell's equator. And in the animation, there you see the X-shaped chromosomes are aligned at the equator of the cell. And I'm hoping you can predict what's going to happen to those chromosomes during anaphase number 2. So now as we look at anaphase number 2, the chromosomes are going to be ripped apart. One chromatid is pulled to the top of the cell, while the other chromatid is pulled to the bottom of the cell. Remember that one chromosome is made from what are called sister chromatids. And so that's what happens during anaphase number 2, is the sister chromatids are pulled to opposite ends of the cell. And as we move on into telophase number 2, cytokinesis is going to divide those 2 cells, there you can see, dividing into 4 cells. The nucleus is going to reform on those 4 new cells. There you see the round nucleus has reformed. One thing I want to mention is that the chromatids are going to uncoil into their loose linear version of DNA called chromatin. So in my animation, the chromatids are uncoiling into chromatin. And the end result are 4 genetically unique haploid cells. First of all, the note in the upper left-hand corner is still there. We started with 6 strands of DNA at the start, but now look at the cell at the top left. It only has 3 strands. The cell in the top right only has 3 strands of DNA. The cell at the bottom left has 3 strands of DNA. The cell at the bottom right has 3 strands of DNA. We started with the cell that had 6 pieces of DNA, and we reduced the number in half to 3. That's what meiosis does. Not only does meiosis make haploid cells, but look at the color of the 3 pieces of chromatin. Each cell contains a little unique mixture of separate DNA because of that event called crossing over that happened way back in prophase number 1. Each of those 4 cells has a different combination of red and black DNA. That's because, again, of crossing over. So before we wrap up this video, I want to mention some of the little differences that occur when meiosis is finished in men versus women, males versus females. Spermatogenesis is kind of the events that take place when meiosis is finished in males. It's kind of how the final touches of a sperm cell are made before the cells are technically able to be used to fertilize an egg. So spermatogenesis is the production of sperm cells. In the picture, we have a diploid cell by the name of a spermatogonium. And this process takes place in male testes. So in male testes, males have millions and millions of these diploid cells called spermatogonium. They're diploid, they have 46 chromosomes, but these spermatogonium are going to undergo meiosis. So after meiosis number 1, that one spermatogonium will go through meiosis number 1 to make 2 cells. And then those 2 cells will go through meiosis number 2, all those notes that we just talked about, to make 4 cells. Now these 4 cells aren't quite sperm cells yet. What spermatogenesis does, again, is make final changes to the sperm cells. You know, males, we release a different set of hormones than females. And so the hormones that males release by our endocrine system helps to shape and change these 4 cells. Through the process called spermatogenesis, eventually the sperm cells will grow tails or flagella. They'll take on a tube-like shape, like a torpedo shape, so they are more hydrodynamic in order to swim in their journey through the female reproductive system. And now again, the picture is just for me being silly. Please don't think that sperm cells have eyes and a smiley face. But you get the idea, I hope, is that 4 cells created by meiosis, they have to undergo through spermatogenesis before they are mature sperm cells able to fertilize an egg. And so this process again makes 4 haploid sperm cells at a time. One diploid spermatogonium will divide into 4 mature sperm cells. Now, for women, the process is a little different, and I believe it's pronounced oogenesis. First of all, oogenesis is defined as the production of the egg cell, or the ovum. Ovum is just the science word for an egg cell, and this takes place in women's ovaries. Women have thousands of cells called oocytes. An oocyte, from the picture you can see, is the diploid cell. Oocytes go through meiosis. Meiosis number 1, that oocyte will divide into 2 cells. But notice how the cell on the left is different than the cell on the right, just in terms of size. That's because during meiosis number 1, when that oocyte goes through meiosis number 1, the cytoplasm is not distributed evenly. The cell on the left received the bulk of the cytoplasm, the bulk of the organelles. The cell on the right, I don't want to say it's empty and hollow, but it's not going to be a functional cell. Those 2 cells, the big cell and the small cell, let's look at the small cell on the right first. The small cell on the right will go through meiosis number 2 to make 2 more cells. Now the big cell on the left will also go through meiosis number 2 to make 2 more cells. So here, as we said in the notes, meiosis makes 4 cells, and there you see 4 cells. But you can see that during meiosis number 2, that big cell didn't divide evenly again. The bigger green cell on the left has the bulk of the cytoplasm, the bulk of the organelles. The other little cell, I don't want to say it's hollow and empty, but it will become a non-functional cell. We call these three non-functional cells, by the way, polar bodies. Now that one large cell on the left, it's not an egg cell yet. It will go through the process called oogenesis and eventually take on the form of a finalized egg cell. Again, women release different sex hormones than men. The hormones released by males allow spermatogenesis to occur. The hormones released by women in their endocrine system allows an egg cell or an ovum to be produced through this final process called oogenesis. Those three other polar bodies, as my notes say, those three other polar bodies simply dissolve. And the one mature ovum can then begin the journey of the menstrual cycle. And so the reason for this picture right here is just a nice little compare and contrast. If you want to pause the video, I'm not going to say much about this, but if you want to pause the video, you can see a nice compare and contrast mitosis on the left versus meiosis on the right. And for those of you who are in my biology class, again, you feel free to pause the video again and try to answer these questions here. If you're in my biology class, pause the video and put your answers on a separate sheet of paper. I'd be happy to meet you before school one day or after school one day and just go over your answers with you and see how you did. So pause the video and good luck.